We got a really nice introduction yesterday into all of the different steps that go into this immunofluorescence protocol. And today we're actually going to be able to sort of see what this imaging actually looks like in real life. And so I am going to, sorry. I don't know the best way to do this. I don't know if I want to see my screen on this computer, but I think I do.
You think you do?
I think I do.
Awesome.
Okay. To start off, we got a lot of really fantastic questions in our feedback form from you all yesterday. And a lot of those questions were really about wanting to know the big picture of these experiments. And that is a fantastic question because the goal is not just to count the number of egg stem cells, but actually the goal is to understand how the number of egg stem cells is determined and the genes and mechanisms and processes that control how many germ stem cells are made. And so, in order to do this, I have to be able to count the number of egg stem cells. And that's what I spent a lot of time talking about with you all yesterday was actually what methods I need to do to count them. But now I'm going to talk a little bit about the experiments that I do to actually understand what controls this process. And so the way that I test this is that I'm interested, I have a list of potential genes that regulate the number of egg stem cells. And so in my experiments, I have two groups of samples for every experiment that I do. I have a control sample where I'm not changing anything about the genotype. I'm not changing anything about the genes that are active in this organism. I'm basically just observing how many egg stem cells are normally in a larval ovary. And then I always have a second group of samples as well, where I've actually taken away one of the functions of one of the genes in the ovary. The term that we use is that I've knocked down the expression of this gene. And basically what I'm looking to see is if getting rid of the expression of one of these genes changes the number of egg stem cells. And so I'm going to actually show you on the screen now, these are images that I took 2 weeks ago that showed a really, really nice phenotype, a really nice example of what it looks like when changing a gene results in more egg stem cells. So this is an image of a control or wild type ovary. And I'm going to actually just bump up the signal a little bit more for you all. And let me do one more thing. So this is kind of the distribution of the number of the egg stem cells in a control sample. And this is the type of image that I can produce when I knock down one of my genes of interest.
And actually, just to rewind a little bit, the cells that are pink are the cells that have vasa in the cytoplasm, and vasa in the cytoplasm is a marker of something being an egg stem cell.
Yep. And so basically what I'm looking for is the number of these pink cells that are lit up in the image. And so it's a little bit hard to tell from these static images, but I think that these two images are a nice comparison. I can put them side by side for you, I think.
Complicated looking software.
Can I? That's unclear if I can. Drive item from images. Well, no. So, okay, sorry. Basically, this is the sort of number of egg stem cells in this one image in a control sample, and then in a sample where I've knocked down the expression of a gene, I see a lot more egg stem cells. And so this was an example of a really successful experiment that was very, very exciting because I was really excited about the possibility that this gene was doing something. And basically what I showed is that by getting rid of the expression of this gene, germ stem cells, egg stem cells increase in number. And so today so that's sort of the big picture goal of these experiments, is to understand what genes control the number of egg stem cells. And so today we're going to be doing some imaging of some new samples that I've been working on this week. And what I'm imaging with you all today is the control. I always do the control first in my experiments, just so that I know that the control samples look really good. Because if there's something wrong with the control, you can't really interpret very much from your experimental samples. So before getting too excited about experimental samples that I then can't end up using, I always image my control first. So hopefully that these images look nice. So the first thing that I'm going to do, and actually Jess, if you would turn the computer really quickly, just to get one shot of what this microscope looks like. So this is the machine that I'm working with here, and it's really big, and it's a very, very powerful microscope.
Oh, Mikala, would you mind stopping screen sharing or pausing screen sharing, and then our faces can be big for the students?
Good idea.
How's that?
Yeah, I got a couple wows in the classroom.
Yeah,
We're used to like our compound microscopes and everything, but seeing something with like all this glassware around it for, sterility and everything. That's so cool.
Yeah. So this is the machine that I am using today, and I'm operating it through this computer. And so I don't really touch the microscope very much. Really, most of it is controlled through the computer. And so I'm going to go back to screen sharing the software that I use, and we're going to be able to start seeing these images come up.
Would it be okay, really quickly, if we look at the sample that's mounted on the microscope?
Yeah, I think it's really hard to see.
Can you see that glass rectangle there?
Yes, we can.
So that is a microscope slide, and the samples are actually sitting on top of that glass slide, and they're sandwiched between another piece of glass. Mm-hmm. And the laser that's going to do this imaging is going to pass through the glass, and then the sample, and then the other side of the glass to collect the image. Perfect. So I'm going to go back to sharing my screen, but before I start operating the microscope and get a little bit distracted, are there any questions about the big picture goal of this experiment?
Yeah, we got a question over here. Are you guys able to hear that?
We were not. If you wouldn't mind restating.
Yeah.
You might have to speak louder or come closer to the microphone.
Yeah, I think so. So there's also a fan running in this room constantly to help cool off the microscope. So this student is, we didn't hear a single word.
Oh, no problem.
Either if they'd come closer, or Mr. Woodbury, if you'd restate.
Yeah.
Is there a human application to like what you're doing? Are you trying to be able to produce stem cells in a lab environment so that you can use it to help heal injured brain cells or injured other injuries, or is what's the application? Is it medical or that's just what I'm wondering.
Yeah.
Or even to rephrase, what is the broader conceptual goal of this?
Yeah.
So I think that there is kind of two different ways to think about the sort of bigger picture goal of my experiments. So one way that I sort of think about applications of my science to human health is that I'm really focused on these very special stem cells that produce eggs for the next generation. And so the work that I do is sort of a way to learn about the fertility of an organism. And in many cases, we care a lot about supporting human fertility so that people can have the kids that they want at the times they want, et cetera. And we really don't have a very good understanding of why some people struggle with infertility. And so one application of this research is to understand how is human health, or what controls egg stem cells in this model system, and how can that be applied to understanding human egg cells, essentially. The other sort of avenue for my research is that the ovary in Drosophila is an organ, just like the human liver or the human heart or the human lung. And we also don't really understand how organs develop from a very small number of cells to a very large number of cells. And so my research is also a fundamental look into how cells in an organ coordinate their growth and their differentiation, so their ability to become specialized types of cells. And so this project is also a really fundamental look at how that process is occurring, because we are interested in understanding how organs form and what is going wrong when they form incorrectly, and potentially how can we even help organs to regenerate themselves. That's another potential avenue for this research. So those are kind of the two different answers.
And I liked what the question asker said in terms of like, can you isolate stem cells from this and grow them in a dish and use them to replace the brain? That is really not what this experiment is about. So we have been able to isolate like human stem cells in a dish and grow them and treat them with hormones and watch them differentiate. And we have determined differentiations for some types of terminally differentiated human cells. But I think one of the things that's really cool, like from my perspective as maybe a bit of an outsider to the science, of studying differentiation in the context of a fly, is something that we don't have down at all for human cells, is thinking about how cells develop in a super complex system where they're surrounded by a bunch of other cells, right? Mikala yesterday was talking about what does an egg stem cell look like versus a non-egg stem cell look like? And something that I think this class brought up, someone in this class brought up was like, the non-egg stem cells, there's ones that look really different from each other, and they were right. We do not have a way currently to recapitulate in a dish a really, really complex environment surrounding stem cells. And that is like a piece of fundamental biology, right? Like you were talking about like the kidney and the heart and like a lot of these, we just like don't yet have the ability to grow them in a dish. People are increasingly looking to grow them in a dish, and the way that they learn how to better recapitulate them in a dish is from fundamental biology experiments like Mikala's that happen in flies.
Yeah, Jess, I really like that point. I appreciate that as a model organism biologist.
And if I'm inaccurate, also tell me.
No, I think that is a really good point. Like, we can take some stem cells out of the human body, and then they do really bizarre things that we don't understand. And oftentimes those experiments where we take stem cells out, they do all kinds of weird things if they don't have their environment around them. And so my experiments, this is a scientific term, we say that they are in vivo, they are in the living organism, and they have all of this context around them. And that is really the best way to study, or at least I would say, as a model organism biologist, that is the best way to understand how an organ develops. So I'm a little bit biased, but there is a lot that can come from model organism biology that I think we don't always appreciate.
And I will say also, like, I'm a very reductionist scientist, so I do what you would call like solving things in vitro. So I really like isolating an individual protein that you can then like grow in bacteria and super purify.
And like so like in vitro meaning like in a test tube.
Really, really in a test tube in a way that is not the type of experimentation that Mikala is doing. But there's constantly crosstalk between our disciplines where like I learn from the things that in vivo people are doing, and in vivo people learn from like my isolation of proteins to better so like there's just constant crosstalk between all of these disciplines, and understanding things at different different levels of how they fundamentally operate then informs application to human health. And I think that's like a really, none of what either of us are doing is like directly applied to human health, but it's still important to understand fundamentally how it works.
Yeah.
And it's also a huge privilege to be able to do that. So thank you for that question. That's like very, very, very important.
Yeah, he gave it 2 thumbs up for that as a thank you.
Awesome. Okay. So I'm gonna get started driving the scope a little bit, and as I'm working, I think I'm gonna sort of turn over a lot of the talking to Jess because I can't really multitask very well. So yeah, let's see what we do.
And keep your questions. There will be points when Mikala's talking more about what she's doing so she can narrate, and then we'll have periods of a break where she's concentrating on figuring stuff out, and then I can help relay questions.
Yeah. And Jess, you can also too like ask me like what am I doing right now, and then I can like
I'm like the MC of this event.
Yeah. Okay.
So actually, Mikala, would you mind telling us what you're working on right now?
Yeah.
If you have the...
So the very first thing that I did was zoom in on the sample so that I'm really only imaging the bounds of the organ more closely. So basically like this here is the entire outline of the organ, but then all of this black space is just the space where the sample isn't, and so I'm not interested in that, and so I like to crop my images really, really tightly, basically as tightly as I can without losing any information, like without cutting off any of the cells. So that's setting the zoom, and then I want to image the entire ovary from the very top to the very bottom, and so what I'm doing here is just setting the top and the bottom, and basically the computer is going to, or the computer and the microscope are going to take an image, move a little bit down, take another image, move down, take another image, move down, and that way I can actually then sort of combine all of those images together to create like a full map of the entire tissue, which is a sphere. And we call this in microscopy, we call this a Z-stack. So if you think about moving around on coordinates in X and Y, that's changing where you are in a 2D plane. And then Z is like the third direction. And so I'm imaging the same place in X and Y, but I'm imaging a stack through Z.
Mm-hmm.
And so I know you mentioned yesterday that this organ is like quite small. These ovaries are, you're imaging like the tip of an ovary.
Yeah.
Or the tip of like a compartment of an ovary even.
Yeah.
What is the distance between each of the images that you're taking?
Yeah, so the distance in between each of the images is a quarter of a micron. So 0.25 of a micron, which is, a micron is 10 to the negative 6 meters.
Yep.
And so then 0.25 would be 2.5 times 10 to the negative 7th meters.
And
So it takes an image, it moves 2.5 times 10 to the negative 7 meters and takes another image.
Mm-hmm.
And it's going to do that 217 times.
And I think I had the conversion, but 0.25 times 10 to the negative 6. Maybe someone in the class can do this conversion than I can. 1/1000th of a centimeter. So
Yeah.
1,000 centimeters.
1/4 of 1/1000th of a centimeter is the distance that it's traveling between each of these images.
Yeah. Which is really small.
Which is,
Yeah.
in part why the machinery of the microscope is so complicated, so it can make these really, really precise changes. Yeah.
Okay.
While you're setting things up, do people have any questions in the class?
Yeah. And so you said this microscope uses lasers, and so those lasers are to shine on those fluorescent tags, and that's how it pops up on the screen.
That's correct.
Yeah, you got it. The fluorescent tags, without going into too much detail, because I know I got really excited about talking about electrons in the second class, the fluorescent tags basically absorb light at a given wavelength. That light loses some energy, and then it gets re-emitted at a second wavelength. So, and that second wavelength is what is being detected. For example, this pink right here, the laser's being shined on at one wavelength. Anywhere where that secondary antibody with the fluorescent dye is present, that fluorescent dye will convert that light into a different wavelength, and then the detector will detect that second wavelength. And so that's where, wherever that secondary antibody with the fluorescent dye is located is where we're capturing the signal.
Yep. And I do have a question. Is it okay if that gets asked?
Of course.
All right, go ahead.
Do they generate a 3D model, or do they just like are they able to
The question is it able to create like a 3D model of this after it goes through everything on the z-axis, or is it just kind of layer by layer? Is my understanding your question? Yeah. Okay.
Yeah, so the final images that we'll be able to look at are not like a 3D model, but I've actually been meaning to for a very long time to use this other software that you can use to essentially compile all of your 2D images into a 3D model and actually like look at what this tissue looks like in 3D.
Mhm.
And one of the things that is really great about the very, very, very thin slices is that because each image is very close to the previous image, it actually is able to create like a very comprehensive 3D model, which is really cool.
And so even though you haven't created a 3D model that like we can look at on a computer and spin around in space,
Yeah.
I do know just from being your friend that you have worked very, very closely with a microscopy specialist to build an algorithm to help you quantify these images, right?
Yeah. So this is something that I can tell you a little bit more about when we're watching one of the channels come in. But for now, I'm going to actually start my acquisition. And while it's running, we can chat a lot more because it's going to run for a pretty long time.
How I'll let you start it actually. Yeah. So we'll come back to that idea of sort of building a 3D model or at least quantifying these images to where you can assign numbers to the cells and how, I mean, neither of us can explain how computer science works, but maybe how like the fact that Mikala's working with an expert in this to help do some more of that quantification. Are there any sort of maybe broader questions that people have that like, I can't answer about Mikala's specific experiment, but I can answer about science in general? Or maybe some specifics I could answer. I can give it my best shot. Oh, you already starting your image?
Yeah.
Oh, great. Well, how about you go back to talking about the person you're collaborating with to do some quantification?
Yeah. So really quickly, so what you're starting to see appear on the screen is just. Can I try turning off the lights? We might look crazy.
Yeah, sorry.
Sorry, but it's actually much better for the imaging if the lights are off. So I prefer to do it this way.
Yeah.
Even if this looks a little weird.
Yeah, we look psycho, sorry.
Yeah.
No problem at all.
Okay, cool. So, so basically I have started the acquisition, and what the computer and the microscope are now working together to do is to image first the DNA in all of the cells. And so this is kind of equivalent to those images that I was using at the very beginning of my presentation yesterday to show you that you can't really get a lot of information from the ovary by just looking at the DNA, but it is really helpful to have pictures of what the DNA looks like because it helps tell you basically where the cells are. So the first thing that the microscope is doing is it's going from the very top of the ovary to the very bottom. It's moving 1/4 of a micron each time, and it's taking an image of what the DNA in the cell looks like, and that's what's coming up in gray. And so the entire image to acquire is going to take about 13 minutes. We're going to be looking at 4 different colors in one image. So about 12 divided by 4, roughly, we're going to be imaging the DNA for about 3 minutes. And then what you're going to see in about another 1.5 minutes is that we're then going to be imaging a different channel. I think the next one that comes up is the traffic jam. So these are going to be the cells that are marked in green, and they're going to be marking the nuclei of some of the non-stem cells. And so then the computer is going to go back, it's going to move the microscope back to the very top of the image, and it's going to take a picture every 1/4 of a micron, but instead it's going to be imaging the green, and then it's going to do the same thing for the red and the same thing for the pink. And at the end, I'll have all of the information, all 4 colors for every single image. So we're going to start to see, and basically I can tell that we've now sort of passed the midpoint of the ovary because now the images are getting smaller and smaller and smaller as you move to the bottom of the sphere. And so pretty soon the microscope's going to make a little noise and it's going to start imaging in green. So basically what Jess is talking about for some of the image analysis that I've been doing is that I want to count these numbers of egg stem cells. Okay, so now it just switched. So now it's imaging green on top of some of the gray. And that is because, again, this traffic jam protein is in the nuclei of some of the non-stem cells. And so it's doing a really nice job. It's lining up the green on top of the gray, but not for all of the cells. So up here, there's no gray.
And so functionally what's happening in terms of the microscope, and I'm actually asking you by a statement, is when it was imaging the DNA, it was shining one laser on, and then collecting the re-emitted light from that laser.
Of one color.
Of one
basically.
laser.
Yeah.
And now it is shining a separate laser on, collecting the information that is re-emitted by the cells and by the fluorophores from that laser. And it is overlaying the 2 basically like re-emitted wavelengths in terms of visually what you're seeing.
Yeah, exactly.
Okay.
And so I actually need to just check 1 or 2 things to make sure that this is working correctly. Basically, the traffic jam green signal should be perfectly aligned on top of the gray because the DNA in the nucleus should be in the nucleus, and the traffic jam protein should also be in the nucleus. And sometimes the microscope makes a little mistake and it puts them in slightly different places. And when it does that, I can't use the images and I have to start over, which is really unfortunate. So basically what I'm gonna do is I'm just gonna turn off the DNA, just look at the traffic jam signal, stop the computer from re-updating the image. So the microscope and the computer are still taking images, but I've just frozen my view of it. And I just want to make sure that
Oh, it's not aligned?
No, but that's for a different reason. So basically then I just want to flash on and off the green. And basically what I'm looking for is like everywhere that there's green, is it roughly, oh, come on.
It is not.
Hold on. It sometimes does this though, if you are asking it to, Okay, so this looks pretty good. So I'll zoom in here. So this, and this are all nuclei. And if I turn on the green channel, these are pretty well aligned. So I'll take this example in particular. This is a really nice circle of DNA signal, and it overlines really nicely with a really nice circle of green. So the reason why it was sort of looking like it wasn't aligned in some of the slices is because sometimes the software, like if you just move too fast, the software will be like showing you. So it said when it looked like it wasn't aligned, it was showing me one slice of the DNA and a different slice of the green signal, and it was just having trouble basically giving me what I wanted that quickly. And so yeah, I've given myself many little heart attacks and then realized like you just have to slow down and let the computer like work. Okay, so now we've switched again, and now we are imaging this HTS protein. This is this red protein that appears around the membrane of non-stem cells and in this very bright dot inside the egg stem cells. And so you can actually see some of these dots really nicely now, where basically I already know that even without my pink signal, that these cells that have the bright dots are gonna be egg stem cells. And everywhere else where you just see the red signal around the membrane, those are non-stem cells. And so this image is really crisp. This one is really nice. It's doing a very good job.
And going back to that broader point of like the ovary being a really complex organ environment and the question that someone asked yesterday, as a reminder, the cells where HTS is around the edge are non-stem cells. And we can see that in some of those non-stem cells, we have traffic jam in the nucleus, that bright green. And in some of those non-stem cells, traffic jam is not in the nucleus. And so we're imaging a bunch of different cell types here, not just egg stem cells and non-stem cells, but many different types of non-stem cells as well.
Yeah. So it's good.
Cool. I was worried you were gonna have to troubleshoot.
I know, me too.
Which happens truly all the time. While Mikala's focusing on something, I will relay. There was one time for a period of a couple of weeks, I think, where she was really, I believe you're pushing the microscope sort of to its limits in terms of taking the thinnest possible slices.
Yeah.
And for a while, things just weren't aligning, right?
Yeah.
And it was like a period of, how many weeks do you think that was?
I was like on and off, but I think it was like probably 6 weeks in total where I just couldn't do any experiment. And it was horrible.
And I think, didn't you realize it was because of the temperature of the room or something?
It was like it was actually sort of a confluence of a lot of different things. And the reason why the problem was so hard to fix was because I actually needed to fix 3 different things before everything would go back to normal. But one of the things that I needed to fix was that when the entire building changed from air conditioning to heat, the temperature of this room accidentally got hard locked at like 65 degrees, which is pretty cold. And then when you start operating this giant microscope, it generates a lot of heat. And so temperature causes glass and metal and lots of things to like expand when they get hot. And so basically the room was heating up so quickly that everything was expanding and moving. And
yeah, especially when you're working with something that sensitive, like any small, like, expansion or whatever, like just totally mess it up.
Yeah, it was, it was bad.
I remember it being bad.
Yeah.
Okay. And then there were 2 other things as well aside from the room temperature.
Exactly. There was something to do with the fact that the microscope slide, the glass slide itself was moving ever so slightly. And then my sample inside the slide was also ever so slightly moving. And so I had to fix all of these problems, and it was frustrating because I would fix one and the problem wouldn't go away. And so I didn't know what was happening. And it wasn't until, I just kept trying different versions of like possibilities that I hit upon one that like worked, and then kind of worked backwards and was like, oh, it must have been the slide, and it must have been the sample, and it must have been the temperature.
Yeah.
So it was a whole thing.
This is honestly a big thing in in like any type of science, not just biology, but like things don't work a lot, and sometimes it's stupid stuff like the temperature of the room and the seasons changing.
Yeah.
And I think being okay with failure and then being really like caring enough about whatever you're doing to like think about all the possibilities as to why something could have failed is deeply important. I didn't feel like I had this skill in high school. I was really a perfectionist and like always had things work right for me. And so I thought in high school I like couldn't do science. And once I started doing science in college, I think the hardest part was just like things going wrong and not thinking that I was a bad person because they were going wrong, but actually being really willing to figure it out.
Yeah, that I feel you 100% on that, especially when it's like, you know, because in high school we want to try to have like an experiment or something where it works and that there's not a whole lot of failure and everything that goes with it, because, you know, we want the kids to see the process and have everything work out. But like, you know, as you said, like a lot of times it might take like 6 weeks for you to figure out like, oh, it's like the temperature and like things are moving and like, you know, of a whole confluence of things that are coming together to, you know, kind of skew results and show off stuff. And so, yeah, I totally 100% feel you on that.
Yeah.
Okay, so now the microscope is almost done acquiring this one image, and it's done, now it's imaging this pink channel, and so it's identifying this Vasa protein that is in the cytoplasm of our egg stem cells. And what I've overlaid here, I'm just showing you the red channel and the pink channel, and I think this really nicely highlights these like bright dots of red inside the pink cytoplasm. So we've now gone through, we are 93% done with this experiment. You can see it's like keeping track down here at the bottom. And what I'm gonna now do is once the image finishes acquiring, I'm going to move to another sample on the scope, reset the top and the bottom of my Z-stack, check the levels of the laser to make sure that everything is like looks good, and then I'm going to start the next acquisition. Are there any questions about the software? Anything that you're observing on this like big panel of software that you want to ask about? Now would be a good time. Now is a great time.
I think I have a question. I'm just trying to like phrase it in my head. Do you foresee like AI helping you out with this process at all? Like either with like helping you like make corrections for like having everything line up, or like saying like counting up like number of cells and kind of pointing some things out with that?
Yeah, it's a really, really good question. I'm gonna drive the microscope a little bit, and then I'm gonna talk about the analysis that I'm doing and how AI definitely could help make the analysis stronger. Be right back.
Yeah, no problem.
Give me 1 sec. I actually want to check the check some of the poll results from people yesterday. But also, yeah, if people have questions, like feel free to ask, and we'll get back to the AI question for Mikala as well.
Cool, no worries.
Sorry.
No problem. And just for timing, we just got about like 15 minutes left.
That's
All right.
perfect.
I know I'm always like cognizant of time, like how much do I have? How much do I have? I'm like always flexibly looking.
Yeah, I'm going to answer a couple of the broad questions from yesterday, which I think people responded to in the poll. One of the questions was like, what are the steps in understanding what is new to research or look for? And I guess I'll just like broadly say, I think I talked about getting involved in research a little bit when I visited. But if you're interested in this, a great place to start researching is if you're in an undergrad in college and like just asking professors if you can work on their research. This is not just true for biology research, but other places. And colleges do have, a lot of times will have like undergraduate departments of research, and you can kind of go there and learn and figure out how to get involved and get started with that. I will also say, I don't know if Jamika wants to speak to her experience, but she's had a sort of a non-traditional research path and started doing research later as she's pursuing a later degree. So I guess also don't feel like you're limited to just doing research in undergrad, but it can be a good place to start. And I want to say that now because I know a lot of people are going there. Got a thumbs up from Jamika. Excellent. But so what are the steps in understanding what is new to research or look for? I do want to sort of talk about like generally how academic research works, which is not the only model of research, but it's sort of the broad, like the primary model of research that exists throughout the world currently. And so I'm also going to speaking specifically to biology. It's different in different fields, but I can explain biology the best. The way that biology research works generally is you have a professor who runs a biology research lab at an academic institution. That's why I was saying reaching out to professors, asking if they're doing research, that is like a really great place to start. And professors will run their research labs and pay for those research labs generally with grants that they acquire through the federal government. So we were talking about funding a little bit yesterday. And again, another reason why the reduction in funding is going to be really challenged for science moving forward in the next few years. But those professors, as they write grants, they will say like, I am interested in addressing X thing, and I have this experience in the field, and like, these are the next questions that I'm thinking about. So on an individual level, when you start thinking about what to research, like operating off of what the professor is studying is like a really good starting point. And professors will usually have at least 10 years of experience in the field, if not more. And so they're just sources of knowledge that can help you understand where to look next. As you go on to becoming if you ever sort of decide to become a graduate student, you'll then again, as a graduate student, work under a professor, and you will again continue to work on science the professor suggests. But especially I think starting as a graduate student is when you start to bring a little bit, and this is true as an undergrad as well, it sort of varies. But as a graduate student, you often work in the field that your professor is operating in, but then bring in new things. Like I think for my professor, he was like, hey, I think this thing would be really cool using these technologies. And then I go off and I do a lot of reading of published scientific papers across fields and disciplines to understand how I can start to apply that knowledge into my research project of interest. I recently heard a Nobel laureate talk on campus. He got the Nobel Prize for studying the mechanisms of pain that happen in the human body. He specifically was interested in studying the receptors that help us, like in our tongues, to sense spiciness, is one of the things that he studied. And one of the things that he talked about in his talk is it can be really, really great to change scientific fields to start to study new research. And I think the reason he said this, right, is like, I talk about professors as sort of these sources of knowledge, and they have experience in their field, and they've built this experience over 20 years, and they really sort of like know the major questions that happen in the field. But when you switch fields, you've built up your own expertise and your way of sort of looking at and viewing the world, and you can then bring that into a new field and sort of apply some of that expertise. So I guess I think I answered it in a very roundabout way, but in terms of like, what are the steps to research or look for, it is like continually gaining experience, continually engaging in like scientific papers that are published, talking to experts in the field who have built up their expertise over 20 years. And also I think entering into a new field with all that newfound experience that you've gained can be a really interesting way to like find new research problems. I don't know if you have any other thoughts on terms of like steps in terms of how do you figure out what to research or what's new to look for?
Yeah, I really like the point about like switching fields. Because I think that can give you like a really new perspective on methods to use to approach the same problem. So like you could, like I say that I'm interested in understanding how egg stem cells develop. And you could think about that from a genetic perspective. What genes do they need? You could think about that from a biochemical perspective. What nutrients or chemicals do they need to proliferate? You could think about that from organ context, like is the organ big enough to support all of the stem cell or all of the egg stem cells? So I think there's like lots of different kind of lenses through which you can view the same question, and that's why coming from different fields can be a really powerful way to do that.
And I will say, Mikala, actually, we were just talking yesterday after doing our first session, and you're going to start to apply like fundamentals of physics to some of your experiments, right? Where you're
my goal, yeah.
Where you're going to be working with physicists and take some of these equations that they use in terms of thinking about like the viscosity of something, like how hard or how soft it is, or like how stretchy it is.
How much tension is it under? because I think that like one of my hypotheses for this system is that like egg stem cells can divide more when the organ is more like under less tension and under less pressure, and that increasing tension and pressure then stops division. And there's some like justification from that from other scientific fields, but I want to start to do some of those experiments in my tissue to see if that contributes.
And stuff like that is precisely where like a fly organ is a great system to interrogate this in, because these issues of like tension are really hard to artificially reproduce.
Yeah.
Mikala, did you have anything that you want to update us on in terms of like what you're looking at?
No, things look good. So my overlap between my two channels looks pretty good.
Great.
Let me see here.
Where does all your information go after you research it? Really important. Publishing papers is kind of like the scientific currency of academia, and it's also where academic research, once you've sort of like looked at a problem enough and from enough different angles to say, I'm pretty confident in saying that this is the answer, you then publish it in scientific journals that are peer reviewed by experts in the field who look at your research and say, well, have you considered this control? Have you considered this control? Have you thought about this? I actually don't believe this point that you're saying here. I want you to go do one more experiment. And once you sort of satisfied your reviewers' asks, the scientific journal will then publish your paper and say, this is peer reviewed, and this is available for actually the general public. Like, this is actually a big fight that's happening in academia right now, where some journals put papers behind a paywall. But increasingly, we're sort of pushing for papers to be published to be free and available to the public and for anyone to access. So your research, once you've looked at it, it goes directly into a scientific journal that then people can look at online.
Yeah.
I've also heard that like you just email the professor who, you know, is doing the research.
Yes.
Yeah, and then they might like be like, oh yeah, here you go, free of charge.
Oh,
yeah,
100%. Professors will absolutely do that.
Yeah.
I think professors feel very strongly that anyone who wants to access their research should be able to access it. And there's this whole weird system of scientific publishing that also exists that puts things behind paywalls. But if you ever find a paper you're interested in, email the professor. Also email the first author. So this is a really weird thing, but basically on papers, usually the professor is sort of like the last author name listed. And then the first author is the person who did the majority of work in the lab. Like what Mikala is doing right now when she publishes her paper, she'll be the first author. Email both of those people, because sometimes the professor isn't always available or checking all their emails, but first authors like us have much more time and are happy to send you a paper.
Awesome.
Yeah. Do you have the bandwidth to get back to the AI question?
Yeah. So in our final couple minutes, I want to talk a little bit about the analysis that I do after I take these images. So the goal of my experiments is to count the number of egg stem cells to determine what genes control that process of making the egg stem cells divide. But there are between like 100 and 200 egg stem cells in every single ovary, and they are really like overlapped on top of each other, and it's like really, really hard to manually count. So when I first started working on this project, I thought that I was just gonna have to sit down and look at every single frame of the image and count how many egg stem cells there were. But it was recommended to me by someone in my lab that this could be a really good place for computer science techniques to actually be able to take in the image and automatically count the number of egg stem cells based on really high expression of the pink signal of magenta. So basically, if I go back to this image that has already been completed, the one that we already took, I feed these images into a computer program that basically correctly identifies this as an egg stem cell and this as an egg stem cell, but won't catch any of these other non-stem cells that are just in gray. And it can do this really, really, really quickly, and it can do it way more accurately than I can. And so this has been a really good investment in this collaboration with this imaging specialist who works in my building. He put together the analysis pipeline for me, and then we worked together to improve the imaging to make the images really clear and crisp, so the computer program could basically see them better. And so we've been working together for about 1.5 years now, and I now have a very nice analysis pipeline that I can run myself. And so I image all of my samples, and then I go up to this really powerful computer that we have in the building, that is actually just a gaming PC, because gaming PCs are they're much more powerful than a normal computer. So you don't even need an actually fancy computer, you just need a gaming PC. But with that, then I can run the analysis program, and within about 30 minutes, it can determine the number of egg stem cells in about 20 samples. And so that's fantastic. That would take me weeks, and I would not be in good shape if I had to just sit in front of a computer for a week and count. And instead, I set my computer and I go get a coffee and I come back and I say, data, how nice.
But it's not quite AI, right?
Yes. So this program doesn't rely on artificial intelligence. But what basically and this gets into a lot of the computer science that I don't quite understand, but essentially artificial intelligence can help train programs that are called neural networks that can do these types of identifications and learning how to identify objects much better. And so one possibility that my collaborator and I have talked about is actually adapting this program to actually use a neural network and actually use artificial intelligence to accomplish this process much faster and much better. But at this point, even non-AI programs are pretty good, and they're also just a little bit easier to run and easier to operate. In order to use this neural network that I'm talking about, you have to spend a lot more time invested in training the neural network. And so I think my collaborator and I decided that this balance of training versus benefit is right, but it's possible that in the future I will transition to using more artificial intelligence in my analysis.
I will say I have very strong feelings about AI and science, which is not that AI is going to solve science. It is a tool to help us do it, and sometimes it's not even the best tool.
Yeah.
I feel very strongly about that. Huh?
I said gotta use the right tool for the right situation.
Totally.
Yeah. I know that we're running a little bit low on time. Is that correct?
Yeah, the bell just rang.
Okay.
Perfect. Well, my image just finished. So thank you guys so much. I hope that it was fun to watch these images. So nice to do this.
Mr. Woodbury, thank you again. Thank you, Jamika, for putting this on. And I will just say, I think when I presented at the class in December, I mentioned this, but if you'd like to reach out and you have any further questions, you will need to go through Mr. Woodbury, but I'm gonna volunteer him and say, feel free to bother him and send us any more questions that you have. Thank you for all of your excellent questions, and sorry we couldn't get to more.
Oh, no problem. Thank you guys so much, and I'll see you guys in about an hour.
See you in an
Awesome.
hour.
Thank you.
Thank
Bye.
you. Bye-bye.